In 2008, Zimbabwe had an outbreak of cholera due to severely underdeveloped sanitation systems that resulted in contaminated water which acted as a vector for the bacteria to respond. Cholera is caused by Vibrio cholerae, which is a gram-negative bacteria that hosts an LPS layer, also known as a lipopolysaccharide layer, which is an endotoxin and will go into depth on how the immune system acts and responds. Over 1.3 million cases a year have been reported due to cholera and are usually caused by ingesting or drinking contaminated water and food that had feces. Once the bacteria enters the body, it travels down the esophagus into the stomach, where the majority are killed off by the acidic pH. The remaining cells will then penetrate the mucus layer of the small intestine and colonize there. Once colonized, the bacterial colonies then release the endotoxin, known as cholera toxin or CT. This is represented by the green Play-Doh strip. CT is what causes the most well-known symptom of cholera, which is severe diarrhea. This is done by CT disrupting the function of intestinal cells by penetrating through its membrane and activating adenyl cyclase. Adenyl cyclase is a protein that conducts a signal transduction pathway and is represented by the red Play-Doh balls. Once activated, it will then lead to elevated CAMP levels that are represented by the purple Play-Doh balls. The ongoing cascade of this activates protein kinase A that signals the opening of ion channels, namely the CFTR, cystic fibrosis transmembrane conductase region. This leads to an increase in chloride secretions, which are notably represented by the yellow Skittles. Consequently, the intestinal cells will begin to secrete fluids that contain necessary electrolytes as well as ions, thus inducing diarrhea due to the water loss. Not only that, the presence of CT disrupts the mucosal membrane of the small intestines, and this then produces a primary innate response of the immune system, as there will be an influx of neutrophils, macrophages, and other phagocytes in the lamina propria. Not only that, but there is also production of cytokines, mostly of interleukins that are proteins to help mediate neutrophil-associated responses, such as interleukin-6, but some include type 1 interferons that are activated in response to toll-like receptor recognition of the LPS. These signals then cause dendritic cells, which are represented by the light green circles, from the Peyer's patches. They will then migrate toward the colonization area and ingest the CT through phagocytosis. Once ingested by this, the antigen fragment will then be cut up and broken down, similar to how a virus's antigen would be, and is then presented by the dendritic cell through what is called an MHC class II molecule. This MHC2 class molecule is represented by a yellow transparent gummy bear that holds a green Skittle for the antigen fragment. The antigen presenting will then cause a helper T cell, which is represented by the dark circle, to latch onto the antigen through the help of an accessory protein called CD4. The antigen presenting cell, as well as the T cell, releases cytokines, causing the helper T cells to mature and activate before then migrating to the lymph nodes. Once there, the activated helper T cells differentiate into two lineages, with one being the cell-mediated response consisting of phagocytes, macrophages, cytotoxic T cells, which is represented by the red circle, through the secretion of cytokines. These cytokines will then activate and signal these cells. Then these CD8 cytotoxic cells will migrate to the colonization area and kill the bacterial cells. This is done through a process in which they will release granzymes and perforin. These are represented by the blue beads and the red fragments respectively. This will then induce apoptosis after the cells have latched onto a receptor that is presenting the CT fragment using an MHC class I molecule that is represented by the red gummy bear. The other lineage for the humoral response consists of B cells that have already encountered the CT and displayed an antigen fragment through an MHC class II molecule, which the activated helper T cells recognize as their antigen-specific receptor. In doing so, the helper T cells activate the B cells through cytokines and cause the proliferation of memory B cells, which are represented by the sticker on the yellow circle, as well as plasma B cells, which release immunoglobulins such as IgA, IgM, and IgG. Because of this, the disease was thus eliminated and remained long-term with memory helper T cells, B cells, and cytotoxic T cells. If contracted again, these will then stimulate memory B and cytotoxic T cells to proliferate into producing activated plasma and cytotoxic T cells once more, without needing the input of the innate immune system. However, for many civilians that are vulnerable in contracting cholera, a preventive measure is to get a vaccine like Vaxchora. This contains a live attenuated version of the bacteria as well as the B subunit of the CT and stimulates the intestinal immune response as it will be recognized as foreign. This will then cause the production of antibodies, thus limiting the colonization of the bacteria if exposed to cholera in the future.